Nice to meet you. My name is Peter Himes. I'm with Microfluidic MEMS Solution. This is the company that's using this inkjet technology for single cell dispensing. What I'll do today is to kind of introduce you to what the microfluidic technology is behind inkjet, how it works, how it can be applied to life sciences, and then with some specific examples for this single cell printing. So let me share my screen. And close this.
We can see your screen, Peter.
Okay, terrific. So once again, my name is Peter Himes. Microfluidic MEMS Solution is the name of the company. So we're going to be talking about inkjet printing microfluidics. It's also called bubble jet and single cell dispensing. Oops. We'll be talking about introduction to MMS, a little bit about the company, and then inkjet for microfluidics, the toolkit that you can develop using this kind of active microfluidics approach. And then we'll be talking about a specific application, in this case, single cell dispensing, what it's used for, different approaches, challenges with traditional techniques, and then how we can apply not just the inkjet technology, but also advanced AI vision technology in order to come up with a robust and really simple to use solution. So the company is based in Kentucky, established in 2016. It has 80 employees and about 6 products. Just a little bit of a time frame. Our founder, Dr. Guan, worked, developed this technology while he was working at Lexmark, based in Lexington also. This was his primary career. So during that time, he was involved with the CMOS MEMS integration and the microfluidic uses in different applications. And this inkjet technology actually goes back to the 70s, I think, is when IBM first began to experiment with it and to be able to integrate MEMS with CMOS in terms of coming up with this technology, which is most commonly used for home printers, as you all know. From 88 to 2002, Lexmark R&D group began to look at this for bio applications. And then in 2012, the company sold its printhead business to Funai, which is a Chinese company. But it kept the microfluidic R&D team for strategic purposes. And it continued to develop that IP portfolio for life sciences in the years that followed. Dr. Guan licensed the IP package or portfolio from Lexmark in 2015, and then established his company in 2016. So MMS is really a company dedicated towards the life science applications of this type of inkjet technology. From 2016, he actually also established an R&D team in Shanghai called Aurafluidics to develop, build, and test the product portfolio. So the same products technology is actually present under 2 different names and based in different regions in the world. This is just an overview of the types of products. All of the products are based on the inkjet cartridges that you can see on the left there, the B150. And it has been used in terms of a handheld bioprinter, a very general purpose biodispenser where you can do tissues and 3D models and spheroids and those kinds of things. And then the single cell dispenser specific products that we'll be introducing today. The Mimi Cup system is a perfusion system that's used with the BP4000 for doing tissue modeling and organoid development, those kinds of things. We're not going to be covering that here, but I'd be glad to answer some questions about it or follow up if there's any specific interest in that. So just a little bit about inkjet. There's actually 2 different versions of inkjet technology that have been developed. The one here is also called bubble jet, in that the CMOS and the MEMS are integrated onto the same chip, and the CMOS drives a heater which goes underneath the channel to create a bubble of air, essentially in very, very fast time frames, typically in microseconds. And that bubble is then what ejects the liquid out of the nozzle, as you can see here in the little animation. Do it one more time. And it's that ejection which gives us actually the control for bio, because the other approach for microfluidics using inkjet uses something called piezoelectric, where you actually have a cantilever based out of piezo material that when it's activated, again, by the CMOS circuitry, it will essentially um, act like a catapult, if you will, and eject the material out through the nozzle with a lot of force. That works great for ink, not so great for bio applications because of the huge amount of shear stress that you're placing on the cells. But having this CMOS and MEMS integration with a unique technology like the bubble jet technology allows us to do different kinds of things. And as you'll see, we have a whole toolkit of different things that allows you to do things like a microfluidics point of care testing chip. This is a diagram of a concept chip. This is not one that we've built, but just as an example. But we call this liquid digitalization for biomedical applications. And what do I mean by that? Well, we're actually ejecting something on the order of 24 picoliters of liquid for each ejection. So that becomes a very small, I guess you can say, quantum of liquid that we can control for the different types of applications that we can do. And this allows us to address different types of biomedical applications. So CMOS MEMS integration leads to digital droplet generation. That coupled with the other integration capabilities that we have can lead to true microfluidic systems integration on a chip, which can lead to biosensing and bioanalysis, which we can add on artificial intelligence image recognition, as we'll also demonstrate today. But liquid digitalization could be, 1D, 2D, or 3D. For 1D generation, this is like micropump and pico droplets. So single molecule detection, digital droplet PCRs, mini bioreactors. Micro droplet generation by using inkjet is very, very efficient. We can generate hundreds of thousands of microbubbles in seconds time. 2D for pico level dispensing is what we're going to be talking about here. This is for both single cell printing and sorting and the organoid or spheroid printing for organs on a chip. This is the range of the products that we have currently on the market today. And then, of course, 3D gets into the areas of tissue engineering, intelligent fabrication, especially when you can have multiple cartridges or print heads doing the 3D printing. Then you can do interesting things like like layers of tissues and more complex tissue modeling. But this type of integration allows us to develop a life sciences toolkit. If you look at conventional microfluidics, you can see that it's really it's a mess to put together. I mean, you're using PDMS, which is very low cost in order to create these systems, but then you're constrained by all of the tubes and the capillaries and the clogging points of clogging that you have to contend with. Whereas with intelligent microfluidics, we actually can take all of these elements and put them onto the same chip. So we can have not just pumps, but also valves, filters, connectors, coolers, heaters, mixers, thermal control, channeling. All of these things then become a toolkit, if you will, for putting together these different types of microfluidics modules. And we have done a number of these different custom modules in the past, so we have this toolkit available to us. But we're going to be talking about kind of like a first step towards understanding this technology today. And that's going to be through these products, the single cell dispensers. There's two different models of it. We'll also be describing them. But first, what are the use cases of single cells? In biologics, cell line development is used for production of antibodies, vaccines, recombinant proteins, also for antibody discovery. For cell therapy, you're looking for clone selection for iPSCs and also for gene-edited cell lines. For gene therapy, you're trying to develop cell line construction for research use and also for viral vector productions. There's a whole area of single cell omics, whether that's transcriptomics, proteomics, or multi-omics, where you need to have single cells in order to build up these research projects. And then for detection reagent development, obviously you're using cell line development for production and detection antibodies for IVD and reagents. And then in synthetic biology, even we use single yeast screening for research and production, which we can also handle with these approaches. But in all these areas of cell line development, where the goal is to create these or generate these stable monoclonal cell lines that can produce high titers of high quality drugs, substances, and production bioreactors. You go through a number of different steps: transfection, selection, clonal screening, cell line characterization, and expansion and evaluation. Whoops, excuse me. I hit the wrong button. And in these processes, single cell manipulation in these critical steps is really, really important for the productivity, the efficiency, and also the quality of the results that come out of it. So there are ways that people do this today, obviously. They all have their own limitations and drawbacks. So FACS is a very common method to do it, but it has a number of drawbacks. Sorter-induced cell stress is a big issue with viability or survivability of the cells during this process. It's difficult to operate. There's lots of clogging, cross-contamination risks, constant adjustments of drop delay and bead calibrations, which means the whole thing is time-consuming. You need to have a lot of starting material in order to have a successful outcome of FACS, and the maintenance with all of the clogging is just is quite high. Microfluidics, another approach, still has shear stress problems causing cell stress. It can compromise clonal recovery. Like FACS, it's easy to get clogged, so it needs constant maintenance. Then the other approach is pipettes, which is based on Poisson distribution-based dispensing. But that leads to poor efficiencies, poor reliability, and super time-consuming to go back and verify exactly which of the well plates has the single cells in it. So being able to combine this bubble jet technology with the other AI intelligence means that we can redefine this single cell isolation into something that's much more elegant. It's fast and efficient single cell dispensing, and we'll be going to more detail as to how this works and demonstrate it for you. We can combine this. We do combine this with AI-powered vision systems, so you can not only detect single cells with high confidence, but also the quality of the cells can be monitored as well, which really increases the quality of the outcomes of the experiments. This leads to assurance of monoclonality. There's no risk of contamination. As you'll see, it's all cartridge-based, so there's no tubing that's involved with the designs. Leads to ease of use, and it's robust and reliable. This is a photomicrograph of the nozzle itself. So you can see the cartridge in the center here, and then the photomicrographs on the left and right. So these are each one of these individual sites are the nozzles on the cartridge. There's actually 640 nozzles total on one of these cartridges. And each one of them is individually controlled by CMOS. So it gives us a lot of control, perfect control over the nature of the operation of the system. This is another diagram. We actually use the center 102 nozzles for the actual inspection and printing. The others are used for reference. Because we have so many nozzles, it actually addresses another concern with these types of approaches, which is clogging. So we essentially have a lot of redundancy built into the nozzles for protection against clogging. But this is essentially what happens. You can see the micrograph here at the top of the individual cells in each of the nozzles that the inspection system inspects. But just as a as a quick diagram. The nozzles containing the single cells are individually printed into the well plates, and then things are refreshed, and then each of the single wells is present in the well plates. Here's a short diagram. You can see the cartridge going in. So this is a simple install, easy to install, and then the solution is piped into the chamber, and then everything else is done automatically. There's setup and priming procedures, but as you can see, the system goes through and does the inspection and then dispensing. Inspection and then dispensing. And then at the end, you can see the single cells in the well plates. So just as a comparison and just some top of the line specs, and we'll go into more details later. I already see some questions coming through. First off, in terms of speed, so it's 2 minutes for a 96 well plate. It's 8 to 10 minutes for a 384 well plate. But single cell seeding efficiency is well over 90%. Efficiency of clone formation, which means the actual survivability and viability of the cells after sorting, is over 80%. This is due to the low stress on the cells. We have single cell imaging and identification capability, which means that not only do we inspect, but we actually give a photographic record of the cells into the well plates as part of the output. It's very robust. So the multi-nozzle design is anti-clogging or protects against clogging. Although this is basically single-use cartridge, if you're doing multiple well plates off of the same type of cell, then a single cartridge can dispense up to 30 or 40 well plates with the single use before it has to be replaced. And of course, free of tubings and valves, no cross-contamination, maintenance-free. So it's a very simple process. You set it up, you go through some priming, self-checks, and automatic calibration of the system. You install the collection devices, you load the cell suspension, everything else is done automatically. And at the end of the day, you actually get a generated report of the dispensing with photomicrographs of each cell that's being dispensed into each well plate. That is possible by the AI system that's built into it. So it's not just a vision inspection. It's vision inspection that's backed up by this AI powered algorithm that's constantly learning for what is a good quality cell. So as you can see on the right, it's not just being able to detect single cells versus multiple cells, but it also can distinguish against debris or impurities. And because of the image learning algorithm that's built into the system, as I said before, good quality cells versus poor quality cells. So you can be assured of only dispensing the quality of cells that you want. This, go back one, talk about this just one more time, because this, this capability is obviously dependent on a specific cell type. And we have a number of different cells that we have already used with our system in order to sort. So these include things like CHO, iPSC, 293, PBMC, T68, and HeLa, and others. If there's other cell types that want to be trained, you want to train the system on that, then that's totally possible as well. It takes a certain number of images in order to do that, but it certainly can be added to the library of cells that it recognizes. So just some examples of some of the performance of the systems. So the deep learning algorithm increases the single cell plating efficiency. This leads to a single cell plating efficiency stably over 90%. This comes from the high resolution image recognition. The colony formation rate is also, very high, very strong. The CHO single cell colony formation rate of greater than 80% is an outcome of the quality of the cells that are dispensed. And that is in part due to the selection of the cells, but also of the low stress on the cells because of the ability to have the bubble jet technology, which we can adjust. So we can adjust the shear strength on the cells, within the machine itself. We've had one of our customers, a top CDMO customer, did this experiment in order to test for monoclonality, and they essentially got greater than 99.5%. I think that was just within the margin of error, but it's essentially 100%. The photo on the bottom right shows you what the system provides in terms of output. So you can see, the cell images or the cell images superimposed on the grid that corresponds to the well plate. I've mentioned both 96 and 384 well plates, but it actually can also be used to print onto microslides. So as you can see in the bottom right, this is an array of cells that's printed on a microslide. The pitch of these cells is much, much smaller than a well plate, obviously. So we have very good dimensional control over the printing and movement platform of the system as it's printing out. So for different types of experiments, this is another approach that we can take. The whole system is designed for contamination-free. This is one of the systems, the larger of the two systems, that has built-in HEPA filters and UV lights. And the cartridge itself, as I said before, is single use. It's designed so that the solution is deposited into the cartridge directly. So there's no tubing, there's no cleaning, there's minimum risks of contamination or cross-contamination with this approach. Of course, it comes with the software in order to set up and to run the system. This is where you would make adjustments in terms of the type of the cell, in terms of other types of parameters that you have. And thanks. So this is what I wanted to just talk about as an introduction. We're going to turn it over to some questions. I see some chats already started. And first question from Felipe is what type of polymers are used to print the circuit? So do you mean the CMOS MEMS cartridge itself?
Hi, Peter. Thank you for the presentation. Yes, well, with the very first slides, you were talking about the microfluidics circuits and how those are printed. So I was interested on that when I pop in the first question.
Yeah, so that's not a polymer. So that's a CMOS MEMS integration. So those chambers for the microfluidics are actually built on top of the CMOS wafer.
Oh, okay, I see.
Yeah, so that's where how you get that CMOS MEMS tight integration. And that's what gives us the individual control over each nozzle head, because the nozzle is printed right on the CMOS circuit.
Perfect. So basically from that and the wafer, you're talking about the nozzle, and that's my following question. The next is the nozzle size. So we're talking about what, like 10 nanometers, 8 nanometers?
Actual nozzle size is about 30 microns. So we can, and that's the nozzle for the ejection of the cell out of the nozzle head, if you will. So that allows us to handle cells in the range of 5 to 25 microns, roughly. And we do work with those different types of cells in that range. We also have some success with yeast cells, which are down in the 3 micron range. Basically the challenge with yeast cells is that it's getting down to the optical resolution of the optical system. So
yes,
yield rate for single cell dispensing on yeast is limited by that. But in terms of the actual print head design, it can certainly handle it.
perfect. And then I have a question around viscosity, what is the nozzle able to work with? But it's all, well, as you know, it's also around the cell that you have there in the system.
Mm-hmm.
Is there any limitation, or are you looking like aqueous systems, or is highly sugary systems for growth?
I am, hold on just a second. I hold on, let me see if I have another spec sheet here on the diagram. Let me just see if I can find that.
And that question comes around the same with the microfluidics that you were talking at the beginning. That traditional systems, you have the shear force, and when you have a high viscosity, you can have a high shear, and you may impact the cells. That's basically why I had the question, was out of curiosity.
Yeah, sure. My my field scientist is not yet on the call, so she would be the one to help answer that question. But you can see here the cell densities that we use.
Uh-huh. Yes.
And like I said, we do successfully use different cell types, CHO, IPSC 293, PBMC, T68, HeLa. So that's a fairly wide variety of different types of cells.
Perfect.
In terms of sterilization method used for the cartridge, so it comes in a sterilized package that you use, that you receive, and then you input it into the MMS machine itself, and then you go through a priming stage in order to prime up the cells, and then after that you're just dispensing the cell suspension directly into the cartridge itself.
Perfect. Perfect.
And then the motion, table within it does all of the, as part of the calibration is to set it up and register it, within the machine and to the well plate. So as I showed in that little video, essentially the cartridge goes over to the camera. The camera inspects those nozzles, figures out which ones it wants to dispense, and then it can go over to the well plates, and it knows exactly which nozzle to deposit in which well plate, and then that is handled automatically by the motion table.
Perfect.
And then you ask, are you forming the colonies inside the dispenser or cultivating in a separate unit? I think that varies case by case. I think in that cloning diagram or example that I showed, which was, go back here. Find the right diagram here. Oh, it was this one. Yes. So yeah, I think this one was done directly in the plate. But we also have, I mentioned we also do a different type of machine for organoid printing.
Yes, you mentioned
And yes, and this is, I'll just show a couple of slides here, because what we do in this case is with this machine, we can dispense, this goes a little bit into how it's done, but here you can see that we're actually dispensing these spheroids, or printing these spheroids, building these spheroids directly into this perfusion cup. So in this case, we do 9 spheres in a single cup. And in that perfusion system is what is used in order to grow the spheroid. So in this case, you have endothelial cells in the bottom, you have your spheroids on the top. It's a pluggable chamber, and then you can have kind of nutrients on the top and drugs on the bottom. This is a system that's designed specifically in order to grow these types of tissue models.
Perfect. Perfect. And the reason why of that question, so I can give you a little bit of a background.
Yeah.
When you're looking into like artificial insemination, there is some air quality preparation. So you are using more than particles with a HEPA filter or something like that. You're actually adding oxidizing agents in the filtration. So the cells are receiving good quality of, or basically pure air. So that's why the question was, is you can keep all of this in your machine, or you need to go and insert in a different unit?
Yeah, fair question. I'll follow up with that with my lab technician on that.
Thank you so much, Peter.
There was another question in terms of commercial availability and business models. So, these machines are all currently on the market, primarily through our Aura division, which is based in Shanghai. And we're in the process of introducing this in the US as well. We have demo systems of the BP4000. This is the unit here that you see, or SCP-4000. That's in the University of Kentucky, and there's also one here on the West Coast, up in Portland. And we showed this at SLAS this past year. We'll be showing it at SLAS in San Diego next year. And we're looking for having a successful launch in the US market, but that's our stage right now. In terms of business model and support, I mean, we're looking for distributors. Obviously, we're looking for customers. We will be providing support, stateside. It is essentially we'll provide all the maintenance for the machine, but it's a fairly robust machine. The main consumable is the cartridge itself. And they're designed for single use, although, like I mentioned before, if you're running many well plates off of the same sample, then you can use the same cartridge with, we think, maybe 30 or 40 well plates. It really depends on the clogging rate of the cartridge itself. Other than that, it should be just continue to work fine.
Got it. Thank you, Peter.
All right, I'm going to turn this off. Any other questions?
No, Peter, thank you so much.
I hope you found this interesting. If you're going to be at BPI, then I will be there. We won't have a booth, but I'll be there to meet with people. And then, as I mentioned, we're also going to be having a booth at SLAS in San Diego next year. So hope to see people there. All right, with that, yeah, I'm going to end the webinar at this point. Really appreciate your time and hope you found it to be of some interest.
Thank you so much, Peter.
All right, bye now.
Bye.